Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 4 and this is lecture 4 of chapter 4. This is going to be the last lecture for chapter 4. Structure of bacterial or prokaryotic cell, and today we will study the internal structure of the bacterial cell. Again, this is a review of the structure of the prokaryotic cell. It consists of external structure, which is appendages and glycocalyx. We already learned this in lecture number 2. And then the cell envelope, right, which is consists of cell wall and the cell membrane. Remember, based on the cell wall, there are two types of bacteria, gram positive and gram negative. Right? We already learned this in lecture number 3. Now today, in lecture number 4, we will learn about internal structure of prokaryotic cell, which is cytoplasm, chromosome, plasmid, ribosome, inclusion and granule, cytoskeleton, and endospores. Okay, remember from this internal structure, the one that have similarity with the eukaryotic cells are the cytoplasm. So all cell will have cytoplasm, and then chromosomes. Okay, all cell will have chromosome and ribosome over here. Okay. So all cell will have this cytoplasm, chromosome, ribosome. Plasmid is very special in bacterial cell, including inclusion and granules. They are also present in bacterial cell. Cytoskeleton is actually also present in eukaryotic cell. And this one is really special because not all bacteria have this endospore. Only certain bacteria like Bacillus bacteria and Clostridium bacteria have the endospore. So this is a special structure of some type of bacteria. Okay, we start with the cell cytoplasm. So cytoplasm is actually the inside or the contents of the cell, which is majority is actually water. So 70 to 80% of cytoplasm is actually liquid or water. And there are some other molecules like a sugar, amino acid, salt, will be also present in the cytoplasm. But again, the main molecules is water. And then chromosomes. A chromosome is actually the one that carry the genetic material, which is the DNA. So the chromosome of bacteria is single, kind of circular chromosome with double helix DNA. So yes, remember DNA is look like this. It has double stranded, double stranded of nucleotide. So this is called what the double helix or double stranded DNA. So this is the one that carry the gene or genetic material. So bacteria has this chromosome with DNA. And then there is another genetic materials that present in bacteria, and this is only present in bacteria, which is called the plasmid. This plasmid actually smaller than the chromosome, is also circular with DNA over here. So plasmid also carry genetic material, mostly for resistant gene. Okay, so carry the resistant gene. Ribosome, again, all cell will have ribosome, including this bacteria or prokaryotic cell. The difference between ribosome in prokaryotic and eukaryotic cell are the size. So ribosome of prokaryotic cell is smaller, which is 70S. That's the size unit. While for the eukaryotic cell, it has 90S. So eukaryotic cell has bigger ribosomes. The main function of ribosome is for making protein. So this is the site for protein synthesis. Now the spatial structure inside the bacterial cell is called the inclusion and granules. So this mostly is used for storage, okay? Storage of molecule, like extra molecules, maybe carbohydrate, maybe lipids will be stored in this inclusion and granules. So whenever the condition is not favorable, for example, there is not enough nutrient, then they can use the storage of food in this inclusion and granules. The cytoskeleton, this is skeleton of the cell. Okay, so skeleton, cyto means cell. Okay, skeleton is the skeleton. So this is the skeleton of the cell, which is consists of network of thin proteins. Sometimes it's called the filament. Okay, that present inside the cell. And the last one, which is the special structure of only some bacteria that have this endospore, which is a group of Clostridium bacteria, Bacillus bacteria. This is the two main bacteria that have endospore, and also there is another group of bacteria called the Sporocercina that also have the endospore. So bacteria that produce endospore, they will have two life cycle, which is they will have vegetative cell. Okay, so during the normal condition, okay, so normal condition, they will have the vegetative cycle. So this is just a regular type of cell where they reproduce. Yeah? But in a very, very difficult situation, like for example, very dry or very hot, these bacteria are able to produce what we call the endospores. This endospore will protect them against any adverse environmental condition. Even they are able to stay in that condition for millions of years. So endospore actually can protect them for a long, long time period during the difficult conditions of their environment. So it's very long survival protection with the endospore. Again, this is the cycle. During normal condition, they will in the form of vegetative cell, which is this is the normal cell in normal condition. So during this time, they're able to reproduce and etc. But if the condition is not good, okay, if a very, very difficult, maybe very dry or very cold, for example, then they start to making this cycle, which is producing the endospore. So this is the endospore over here. If you see, the cell is actually really, really, is the cell over here protected by a very thick layer that protect the cell from any bad condition of weather or environment. So when the condition is favorable again, then they can hatch into vegetative cell. Okay. So again, in this condition, they can stay for a very long period. As I mentioned before, it could be million year. Okay, even they can stay in this condition for million years. Okay, so this is a special structure of some bacteria, which is endospore. So they have two type of life. Okay, so which is the vegetative type and the endospore type. This endospore actually will cause them to be very difficult to destroy, to be destroyed. So when we try to kill this bacteria like Clostridium or Bacillus bacteria, then we have to perform a process is called the sterilization. Okay, so this sterilization will be able to destroy this endospore. Regular cleaning methods like disinfectant, for example, or boiling water will not kill this endospore, will not destroy the endospore. So it needs special methods. It's called the sterilization. Example of sterilization is using autoclave. Okay, in many lab or clinic or hospital, they will have autoclave in order to sterilize their equipment. So this is actually the process that will also kill the, of course, kill the vegetative cell and also destroy the endospores. I think that enough for this lecture. Next, we will continue with the bacterial cell shape and arrangement, including also some classification of bacteria. Goodbye now.